So I'm super, I got it. I'm extremely excited today to get to introduce Dr. Adam Fields. I think I kind of wrangled you in a little while ago when everybody was really interested in cannabis, and you know, you're the man that's been doing a ton of work in this area. But Dr. Fields' research has really been focused on understanding and developing therapies for neurodegenerative diseases. He's been particularly interested in how alterations in mitochondrial function and chronic inflammation interact with neurodegenerative processes. His work includes examining postmortem brain specimens from humans and animal models to identify neuropathological signatures associated with diseases. He uses in vitro models to dissect the neuropathogenic mechanisms and test therapeutic strategies in neurons, astrocytes, and microglia. And ultimately, the goal of his work is to identify therapeutic targets and treatments that will hopefully improve clinical outcomes for people suffering with neurologic disorders. So, super excited to hear about his work and where things sit in the area of HIV, cannabis use, and macrophage phenotypes. So, with that, I'm going to pass the baton to you.
All right. Thank you, Leah. This is the first time to present this, so it could be a little choppy. But most of it is published already. But then we have one that the last study that we'll talk about is in revision. And then we have like a little bonus study at the end. But I just want to stay at the out front. I'm part of a big, amazing group out here. The HIV Neurobehavioral Research Program is kind of our mothership, and so I work with a lot of clinicians and that recruit all the people and make this kind of work possible. And we're really just the part of the basic science end of things to try to figure out the mechanisms that are behind the associations between cannabis use and HIV, macrophage phenotype, and what's going on in the brains of these people. So, all right. So, just an overview. We'll talk a little bit, give a background. HIV, macrophage phenotype, and then a couple of pathways that we are looking at, TREM2, NLRP3, and then immunometabolism, which is basically how the immune system and metabolism are integrated in sort of you can't really change one without the other, right? It takes energy to run the immune system. And if you excite the immune system, it's going to affect metabolic processes. And then cannabis use in people without HIV and people with HIV. And then we'll get to our hypotheses, a little bit about our approach, and of course, the results and next steps. And this is a work in progress. Um, and I'll explain that as we go through. I have to just put this up here. I know I'm probably preaching to the choir, so to speak, and talking about HIV in the brain, but I really just wanted to show this to point out um, we know that HIV infects immune cells, and then sometimes early after infection, the immune cells cross the blood-brain barrier into the brain, and they seed the brain, and then hopefully the people soon are diagnosed with HIV infection and then get on antiretroviral therapy, and the viral loads begin to be suppressed. But there's still these macrophages floating around, and we're really focusing on these monocyte derived macrophages, the monocytes that traffic into tissues like the brain and then differentiate. And they're not necessarily the cells that we're looking at. Honestly, we don't find much virus evidence of infection at all, even after taking them from people with HIV, putting them in a dish, growing them for a week. We don't see transcripts of HIV or HIV proteins. But as I'll show you, the phenotype of these macrophages that they differentiate it's pretty clear that they're different than the macrophages that you get from people without HIV. And that's what we're focusing on in these studies, and how the phenotype of these cells that are floating around are related to the clinical readouts of the people, cognitive impairment or depression or biomarkers or brain imaging. We won't get into all that, but that's how this study is positioned, which I'll explain more. So macrophage phenotype. I'm kind of repeating things here, but I'll get a little bit into how the macrophage phenotype is characterized. But our evidence suggests we're looking at bystander macrophages that we're taking the blood from people and with HIV and without HIV, but the cells that we don't see much infection. So we differentiate them in addition and look at their phenotype. The people we're getting the blood from they're virally suppressed for the most part. And we think, there's evidence that there's still inflammation in virally suppressed and viremic people with HIV, which is what we think is leading to some of the neurocognitive impairment in these people and general brain dysfunction or damage in the people. And you have several types of macrophages. You've got the resident macrophages and infiltrating monocytes that differentiate into macrophages. And that's really what we're looking at. Although we think that it's possible that the phenotype of these cells at least somewhat reflects what you're seeing or what is happening in the resident macrophages. So we aren't really testing that here. So I put this picture up, and I'm sure it might offend some of the more macrophage biology experts out there, but it's, putting this dichotomy of M1 versus M2. And I think it's much more complex in that than having just these two types of macrophages in the body. But really, I think it just gives us a picture that macrophages tend to skew towards this M1, which is more inflammatory. They get their energy more from glycolysis, and they produce more inflammatory cytokines and proteins versus the M2 leaning macrophages that are run on mitochondrial function, oxidative phosphorylation, and they produce more anti-inflammatory cytokines, more of that homeostatic phenotype. And the general idea is that in people with HIV, where you do see higher levels of inflammatory cytokines and neurological dysfunction, more of the macrophages may be of this M1 phenotype. And if we could skew them back to M2 phenotype, maybe that would be neuroprotective. There are a lot of other immune cells that are important as well. We aren't studying them here, but we understand that it's not all about macrophages. But our data do suggest that what is happening in macrophages is reflecting what's happening in the brain, at least in the clinical readouts. So that's why we think it's at least a good place to start. So is cannabis use good or bad for the brain? And the answer is yes. It's probably both, depending on the context and in also the dose that people are using and maybe how they are using it, and then which kind of cannabis they're using. And so that's basically what this graph kind of shows. You've got a few curves where you have health effect on the y-axis and the dose on the x-axis. And you could imagine these lines represent three different people. For one person, their optimal dose may be way over here. It may be very low on the left. And that may depend on a bunch of different things like their state of health and how they're using and how often they're using it. And then this, I guess, sort of average person, so to speak, the optimum dose might be here. And for somebody else, it may be way up here. But if you use too much of it, like most things, it could possibly be toxic. And so that's the running hypothesis, not just in people with HIV. I think also in things like age-related disorders, where it's being tested in mice. And there was actually a study, I read one in Brazil recently, where low doses of cannabis appear to slow the effects of aging-related dementia in a big cohort of people. So there seems to be this hormetic effect that I want to really get clear across. We don't think that just using cannabis is good for everybody. And that's because there are clearly detrimental effects of cannabis, like impaired cognitive function. If people start using it too early, it may affect brain development, including hippocampal and cortical volume, leading to mental health issues. There's GI tract issues associated with these really strong forms of cannabis. And in healthy people, and we'll show you some evidence of this, It may impair immune responses, which the immune system gets a bad name a lot of times, but really a properly regulated immune system is what we need, right? We need an immune system that will respond to infections uh, and quickly and put an end to it. We just don't want it to continue on and turn into a chronic immune response, which is what it looks like is going on in people with HIV. So, why is cannabis use important in people with HIV? Why is it important to study this? Well, they have increased inflammation generally, but people with HIV use cannabis at a higher rate than the general population. I'll show you some results of some studies from other people, and there's some, I couldn't mention them all, but there's evidence of lower inflammation and better cognitive outcomes in people with HIV that are using cannabis compared to people with HIV not using. Macrophages mediate inflammation, and there is evidence, other studies that suggest that cannabinoid receptor agonists and other things may be able to reduce or modulate the immunometabolic mechanisms in macrophages. So some of the studies, these are out of our group. Basically, this first study here, they on the left side, it's showing a schematic from a paper by Ron Ellis, and they basically found that there were lower levels of these inflammatory cytokines in the CSF of people that had used cannabis recently, people with HIV. And they posited the model that if you add HIV to these resident brain macrophages called microglia, you get more cytokines and more inflammation. But if cannabis is on the scene, it may reduce the number of cytokines. And then another study about a year later showed that this MCP1 is lower in CSF of daily cannabis users compared to non-cannabis users. And so that both suggest, and these are cross-sectional studies, but they suggest that that cannabis use is at least associated with lower inflammatory signaling. Now, it doesn't show, a cause and effect, which is important. But I also want to point out that if you look at even the people with HIV not using cannabis, the lowest levels of MCP1 are in an HIV positive, maybe two people here down here. They're HIV positive non-cannabis users, but so are the highest levels. And my point is that it's not, the people aren't the same. And so because this average is higher than people without HIV, that doesn't mean that everybody has the same disease pathogenesis. And so what we think by taking the samples from the patients is in looking at their cells, maybe we can find the people with higher inflammation and figure out if they could benefit from targeting the endocannabinoid system. And maybe these people down here, maybe they just don't have as much inflammation, and maybe there's something else driving some sort of disease characteristics that they might have. So my main point is we need to pay attention to the spread and not just the average, because we may be able to figure out things about individuals by using their cells and how they respond to stimuli. But the average MCP1 and CSF of daily cannabis users was significantly lower than the average of the non-cannabis users. So that does suggest that it could be anti-inflammatory in people with HIV. And then this was another study that got more at brain function, some things that I don't fully understand, but it was fairly recent last year. But they found, I took this quote from the abstract, longer duration of time since HIV diagnosis was associated with a less efficient inhibitory processing among people with HIV who did not use cannabis, but not among people with HIV who regularly use cannabis. And then they put this graphic in the manuscript. And again, this pink is HIV negative people. So they have high levels of this oscillatory gamma, which again, don't ask me to tell you exactly what that means. But when they use cannabis, these levels go down, equivalent to people who have HIV and aren't using cannabis. But people with HIV using cannabis, it goes back up. And I point this out because You're going to see a lot of that today, where we see an effect of cannabis use in people with HIV or people without HIV is somewhat similar to what you see in people with HIV not using cannabis, and it's reversed when they are using cannabis. But again, it's not everybody. So I think a big point is I'll probably repeat this ad nauseum, but we want to find the people. We want to find ways to find the people who could benefit from such therapies, not necessarily psychotropic cannabinoids, but cannabinoid receptor agonists or some way to modulate macrophage phenotype. And it's not all good news for people with HIV. This is a recent study that showed people using cannabis have lower microbial diversity of their microbiome, which is not a good thing. It's associated with brain function and immune function. So on the y-axis, you have this diversity index of microbiome. And on the x-axis, you have this THC use per day, and you can see it goes from to the bottom right. But again, some of the highest diversity is over here on the right, where they're using a lot of cannabis. So maybe cannabis is affecting these people a lot more than it is these people over here, or these people here. So again, I just wanted to point that out. And there's other studies that suggest cannabis use could be associated with some measures of neurocognitive impairment. It's associated with cardiovascular risk and less imperfect art adherence. So all those things are good to keep in mind. So I probably didn't need this other than to just restate why we are looking at macrophages and cannabis in people with HIV. The idea is that HIV is inducing, whether it's infecting the cells or it is infected, or the cells that it does infect are producing certain cytokines or chemokines that are affecting the bystander cells. We hypothesize that the macrophages are shifted toward this M1 phenotype, which is higher glycolysis, less mitochondrial activity, and more inflammatory gene expression. And that will play out by less TREM2 expression, more NLRP3, and less mitochondrial activity. And I will say, this is one of my first studies, our first studies, that the hypotheses sort of, for the most part, are kind of not confirmed, but at least they aren't contradicted by the data that we have so far. So that, I don't think I have to say this, but just to let you know, that's the overall hypothesis. Again, appropriate doses of cannabis or certain cannabinoids are anti-inflammatory and neuroprotective in some people with HIV. So we broke it down into 3 studies because this has been going on for about 6-7 years now. And so we kind of published as we have been going, but fortunately the data are continuing to support each other, which is also not something that always happens. But the first one, cannabis use is associated with increased TREM2 expression in macrophages from some people with HIV. And then cannabis use is associated with reduced NLRP3 related gene expression in macrophages. And then the last one is cannabis use will promote anti-inflammatory immunometabolic phenotype. So this is an overview of the study. And just to point out how many people it takes, I'm really focusing down here on this AIM-3. And this is one of 2 studies that are led by Dr. Jennifer Udicello. She is a neuropsychologist here at UCSD, and we've been collaborating for a long time. And the studies are just amazing, and we're very fortunate to be a part of it. But basically, it's the HNRP, a big group of people, recruits people, analyzes them, takes blood, does imaging, lumbar punctures, doing all, looking at biomarkers, all the cognitive testing. And then the day that they do that, they give us the blood. They give some of it to Dr. Marcondes and then some to us, and we pull out the cells, different, or at least culture the cells that we are interested in. We have started keeping all the other cells as well, but we are interested in the monocyte-derived macrophages. And in this particular study, we analyze the mitochondrial activity and inflammatory phenotype of the monocyte-derived macrophages. We generate conditioned media by treating them with stimuli that are relevant to HIV, like HIV, IL-1 beta, cannabinoids. We generate conditioned media and then expose astrocytes and endothelial cells to that conditioned media to understand how it affects the blood-brain barrier and how it could be different for people with HIV using different amounts of cannabis. For this project, or what I'm going to show today, we're only focusing on monocyte-derived macrophages. But just to give you an idea, this is all coming together, and there will be a lot more to come. And we're pretty excited about it. But to get a little more granular, what we are doing, and it's all from this study, but they recruit the people, they break them down by HIV status, either with or without HIV, and then you have what we're calling naive, which means they haven't used in the last year, moderate, which is 1 to 6 days per week of cannabis use, and then daily, they use it every day. Now, those are not perfect classifications. I don't think there are any perfect classifications, but that's where we started. And sometimes we see effects that are based on those. And then other times we group them together as either cannabis use versus no cannabis use. So we receive the blood in the lab. We culture MDM cells, monocyte-derived macrophages, by plating the PBMCs. We treat them with stimuli like IL-1 beta to mimic inflammation, and THC or cannabidiol. And then we look at things like mitochondrial markers, looking at these potentiometric dyes that light up active mitochondria. We look at RNA, we look at protein in the supernatants and in the cells. And then we have run some seahorse. One limitation of this is you can only take so much blood from somebody in a day, right? So you can't do everything in everybody, and you get different numbers of cells. But we generally have RNA from everybody. We have supernatants from everybody. We have mitochondrial readouts from most people. We do not have seahorse from all of them, though I wish we did. It's also very expensive to run. But this is the whole idea. So what I'm going to be showing you now are data from plates of cultured macrophages that we received from these people that are stratified by cannabis use and HIV. And then we sometimes it's just the baseline, and it'll be like vehicle treated macrophages and comparing them between the clinical groups. Other times I'm going to show you where we treated the macrophages with IL-1 beta or cannabinoids to see how they respond compared to their baseline, because we think their response to a stimuli reflects their phenotype, right? How reactive are they to certain stimuli? So I'll repeat that, but hopefully it makes sense. All right, so first we're going to talk about TREM2. TREM2 is a receptor on macrophages. It really got famous first in cancer, which I didn't know until one of my students let me know that a couple of years ago. I heard about it because TREM2 has a mutation that predisposes for Alzheimer's disease. And no one had really looked at it in HIV. And there was this whole idea that people with HIV may be aging prematurely, and there was good evidence for it. So in 2018, we looked at TREM2 in the brains of people with HIV we get the brains from the National NeuroAIDS Tissue Consortium. And we did find that there were lower levels of TREM2 on microglia in brain tissues from people with HIV with cognitive impairment. A couple months later, Gislin et al. Showed that elevated levels of soluble TREM2 in the CSF of people with HIV with cognitive impairment. So that was always nice to find. And this is important because TREM2 is a receptor that it recognizes amyloid beta plaques. It binds to ApoE, so it can phagocytose and clear the brain of dying cells and amyloid plaques. It basically is like the trash truck of the brain. If it's not functioning, your brain can get clogged up with things. So our hypothesis was that cannabis will promote TREM2 homeostasis and function in macrophages of some people with HIV. So these data are RNA from monocyte-derived macrophages, just vehicle treated. So basically the baseline RNA, and you have 4 groups in the 4 different panels: HIV negative, cannabis negative in the top left, HIV negative using cannabis, HIV positive cannabis negative in the bottom left, and then in D, HIV positive, cannabis positive. And this was a couple years ago that we didn't, we were recruiting people so that the numbers are pretty low. But in the HIV negative cannabis group, you see a very strong relationship between TREM2 RNA levels on the y-axis and age, where TREM2 levels are increased or mRNA levels are increased with age in monocyte-derived macrophages, which we thought makes sense. But when they use cannabis, or at least in people using cannabis, this relationship is not it's not as strong, although it is still up and to the right. But in people with HIV, there was not a significant relationship. But if there was a relationship, albeit not significant, it was opposite in direction than people without HIV. But if they use cannabis, the relationship just got stronger, and that was actually pretty significant. This wasn't really consistent with our hypothesis, but we have other things to look at. So next, we look at TREM2 related genes in groups, and basically the control is HIV negative, and then we have HIV. I'm sorry, this turned out blurry. I just cropped it from the paper. Didn't do real well. But so the white bars are control, which is HIV negative naive, and then you have HIV positive naive in gray, and the darker it gets, that's HIV positive moderate users, HIV positive daily users. But as I showed you earlier, what I wanted to point out, and these are all genes that are related to TREM2 function and pathway. But what I wanted to point out is in HIV negative, you have your level. We basically compared the levels to the HIV negative group. In HIV positive, CHIT1, for example, went down, but when they use cannabis or people using cannabis, the levels are elevated again. And you see this pattern, you'll see it quite often in the data. So it's supporting that cannabis, HIV is affecting people in one way, and then cannabis use seems to be reversing that effect with regard to TREM2 pathway. So then we wanted to look at the protein in the macrophages. And these are macrophages. We weren't able to do this in everyone, so it isn't stratified by HIV status or cannabis use. We're doing that now. But if you look at we took the macrophages and we treated them with IL-1 beta or CBD plus IL-1 beta, and then we fixed the cells and we immunostained them for TREM2. And what you can see is in the vehicle treated, you see a good amount of TREM2, nice staining. And then whenever you treat them with an inflammatory cytokine like IL-1 beta, the levels go down. CBD, they actually not changed much compared to vehicle, but whenever you treat CBD plus IL-1 beta, it reverses what the IL-1 beta did to some extent. And that's in the membrane-bound TREM2, the TREM2 that's actually on the cells, but also in the soluble TREM2. This is the TREM2 that is not attached to the cells, so it's floating in the supernatant. You see the reverse effect, where IL-1 beta increases TREM2 in the supernatant, but CBD didn't do anything significant, but it did reduce the amount of TREM2 that was in the supernatant, suggesting that CBD may promote TREM2 attachment and possibly function on the macrophages. We're looking at phagocytosis now in the macrophages. And so these data are actually more recent, and we're revising the paper that is included with, but I think these are a lot more numbers. I think it ends up being like 100 different people. But what we did here is we measured soluble TREM2 in the plasma of people without HIV using different amounts of cannabis. So if you look at panel B, you have soluble TREM2 on the y-axis, and then on the x-axis, you have people without HIV and people with HIV, and they're stratified into the three groups of cannabis use. People without HIV, the non-users have the highest level of TREM2 in the plasma. But as they use cannabis, it goes down. People with HIV have the highest average levels of TREM2 in the plasma. And this is consistent, I think, with data that we published and also Gislin in the CSF. But you see high levels of TREM2, and as they use cannabis, it goes down. But again, you see a spread, right? Like in the non-using group, there's as far as TRIM2 goes, there's clearly two different populations of these people with HIV, but this is consistent with the other data that we show in a lot more people. And then if you correlate these soluble TRIM2 plasma levels with global deficit score, where the higher number is that they're performing worse cognitively, the higher levels of soluble TRIM2, the worse their cognitive scores. And most of these are in people with HIV who are naive. So this is all supporting a role for TRIM2 and cannabis and possibly affecting TRIM2 function, which would, we believe, affect the macrophages, which I don't think I need to say much about the conclusion. So I kind of covered that pretty well. But what we don't know is, does cannabis provide neuroprotection in some people living with HIV by promoting TRIM2 function, and if so, by what mechanisms? And we really are limited because we need, you need longitudinal studies. You almost need like a clinical trial, but we're not proposing that. I'm just saying at some point when you find maybe a cannabinoid receptor or a cannabinoid or a group, a combination of cannabinoids or terpenes that do promote TRIM2, there's more granular studies that need to be done. Oh, I'm going to keep going because I want to make sure we get through all of it and have time to talk. The next study is looking at NLRP3. NLRP3, you may or may not have heard of it, but it's a multi-protein complex that forms whenever you have these pathogen-associated or damage-associated molecular patterns. And the first part of it in the schematic is priming, and I don't think I gave anybody credit for the schematic. It's not mine. I do apologize for that, but I thought it was very nice. But the first part is priming, which is basically where you have to make sure all the components of the inflammasome are in the cell so that whenever you have this two-hit like HIV or mitochondrial damage or some insult, the proteins can complex and aggregate, which allows processing of these pro-cytokines like IL-1 beta and IL-18 and gasdermin D, for example, which then get cleaved by caspase-1, and then they can be secreted into outside of the cell where they can bind to receptors on other cells and induce the proper amount of inflammation, hopefully. The problem with HIV, or the running hypothesis with HIV based on a lot of good evidence, is that this inflammasome is chronically activated, and it's leading to the inflammation, the low levels of inflammation that you see in people with HIV, even when they're virally suppressed. And there's a number of studies that suggest this is happening. So we thought that cannabis use, if that is happening, and cannabis use is associated with lower levels of inflammation in people with HIV, then maybe it's acting through the NLRP3 inflammasome. There is a drawback that we don't really get enough cells to do protein work in all of these patients. But anyway, I'll show you what we did. We can do some protein work. So the first thing we did was just we're looking in the monocytes for NLRP3, mRNA, and related RNAs like IL-1 beta and IL-18. This first graph is the same graph as this one. We just published this one on the right. Bryant Avalos, Dr. Avalos led this in our lab. But I showed this one in this form because it shows all the dots, which we probably should have done in the paper. So each dot represents RNA levels in monocyte-derived macrophage culture from one patient. And my point is just, again, although the average of NLRP3 is much higher, like twice as high in people with HIV, the lowest NLRP3 is also in macrophages from people with HIV, but so is the highest. So again, it's just to point out there that not everybody's going to be the same. So you see it's a significant increase in average level of NLRP3 in the MDMs from people with HIV. There was not a significant increase or in IL-1 beta, but it is increased by close to probably 40%. IL-18, we really didn't see much of a difference. But when we went and we treated the cells with IL-1 beta, CBD, or CBD plus IL-1 beta, what I wanted to point out here was really with NLRP3, you basically see a greater increase in NLRP3 RNA in the people with HIV. I'm looking at this top left panel here. And you actually, CBD, it seems to reduce it quite nicely in the people without HIV, the NLRP3 levels. IL-1 beta levels, so far, we really haven't seen, and I'm looking at D below on the left side, we really haven't seen much of a difference in how the MDMs respond. But with IL-18, the difference become more pronounced whenever we further stratify the MDMs by cannabis use patterns. So now we'll move over to the right side of the slide. Now that I'm doing this, this is really just too busy of a slide. But let me try to point out, I just want to kind of point you to the highlights. If you go to the right panel of graphs, the NLRP3 fold chains, the left side, the top left graph here is in people without HIV, and then it's stratified by different cannabis use patterns. You can see we don't have as many as we'd like for all of them. But overall, you don't see much difference, except moderate might have a more effect against IL-1 beta and CBD, but it's only 3 per group. In the people with HIV, just overall NLRP3 levels are higher in response to IL-1 beta, CBD, and CBD plus IL-1 beta compared to people without HIV. IL-1 beta, we don't see a lot of difference, but IL-18, it gets kind of interesting in that now we're at the very bottom of the slide, H and I, IL-18 fold change. You can see in people without HIV, you have the naive, moderate, and daily users. When you treat with CBD plus IL-1 beta, you get this almost stair step of higher levels of IL-18. But the opposite is true in people with HIV, whereas the IL-18 is stair stepping down in the groups with more cannabis use. We don't know exactly what it means, but what it does suggest that cannabis use is affecting macrophage phenotype differently in people with HIV than people without HIV. And it's also affecting IL-18 different than IL-1 beta. So the reviewers asked us to look at IL-18 in the supernatant of the monocyte-derived macrophages. Probably should have done it anyway. We can't look at IL-1 beta because we're actually treating the cells with IL-1 beta. I wish we could. But what we found, so these graphs are from ELISAs for IL-18. And in the top left, you have people without HIV looking at IL-18, and you really don't see a difference between sorry, one second. Yeah, the naive versus cannabis users. And then when you break down the cannabis users into naive, moderate, or daily, you really don't see a difference. But when you look at the people with HIV, the cannabis users have drastically reduced amounts of IL-18 in the supernatant. And it seems to be whether they're moderate or daily. Again, we don't know exactly what it means, but it does suggest that there is a difference in phenotype of these macrophages with regard to NLRP3. And if IL-18 is promoting inflammation with people with HIV, this could be a mechanism through which cannabis reduces that inflammatory phenotype. And that's basically what we say here. We don't know yet if NLRP3 is oligomerizing, sorry, I misspelled that, and are the MDMs from each group more or less inflammatory or neurotoxic. So we're doing things now like taking the same condition media and putting it on astrocytes and endothelial cells to see the effect on the blood-brain barrier, but also neurons to see if it's more toxic and if the toxicity correlates with global deficit scores and other readouts. Okay, taking too long. So now we're going to look at immunometabolism. Again, I'll just remind you that the more inflammatory phenotype has higher glycolysis and higher levels of inflammatory cytokine expression. The more anti-inflammatory or M2 leaning phenotype has more mitochondrial activity and less inflammatory gene expression. And so we hypothesize that cannabis use will promote an anti-inflammatory immunometabolic phenotype in the macrophages. And the very first thing I'm going to show you is this basically confirms things that were previously published. The left panel does. And you have global deficit score on the y-axis, and you have stratification by people without HIV or people with HIV, and then we further stratified by cannabis use. In people without HIV, the more cannabis they use, the worse on average their cognitive score, which, I mean, it kind of supports maybe you shouldn't use cannabis, unless you have a good reason. But people with HIV, if they're not using cannabis, they have the worst cognitive scores of all, and within that group. And the more cannabis or the people using more cannabis have lower average cognitive scores. Although it's not all of them, you can see there's a group where they're performing better than the rest of the group. And just so I'm clear, higher global deficit score means worse cognitive score. So in the right panel, in these same people, we measured mitochondrial signal. And what you see is the more cannabis they use, the higher the signal of mitotracker in these MDMs, suggesting that their macrophages are using, their mitochondria in the macrophages are more active in the people that use more cannabis. And this is also true with people without HIV, despite the fact that their cognitive scores are getting worse. But at least it is consistent with our hypothesis. I will point out, it does not show that cannabis is doing anything. This is cross-sectional. It doesn't show that cannabis is improving deficit score or improving the mitochondrial activity. What we tried to do to get at that is, in this year, we have, I'm looking at the left graph, but they're all pretty much the same. You have mitochondrial signal in the monocyte-derived macrophages on the y-axis, and then different measures of cognitive function on the x-axis. And these are monocyte-derived macrophages that were treated with THC. And what you find is the people who are performing the best cognitively, their macrophages respond to THC with greater mitochondrial activity. Saying that another way, THC induces greater mitochondrial activity in monocyte-derived macrophages from people with better cognitive scores. It's not proving anything except just what I said. It's not proving anything in the people, but it is consistent with the idea that cannabinoids or cannabis is improving, it's promoting this M2 leaning phenotype, and that is at least related to better cognitive scores. Again, we would need like clinical trials and longitudinal studies to prove any of this. But I think the way you could do it is take their cells, try to figure out who would be responsive to the cannabis, and pick those people to try a cannabinoid receptor agonist. So next we want to look at the genes, and basically these are the genes in the monocyte-derived macrophages. We looked at, and Dr. Halcrow from our lab did a lot of this. And I should have said the NLRP3 study was really led by Dr. Avalos and a new student in our lab, Kyle Walter. This one was led a lot by Dr. Halcrow. He helped us wrap it up, and Mary Ford in our lab. But all these data, we've been working on them for years. So it's kind of snowballing. And we have a lot more to come. But this is basically telling the same story that I had, that us and others have been telling. Whereas this first graph is looking at a series of glycolytic and oxidative phosphorylation genes. And if you compare, you have the fold change on the left. If you compare people without HIV daily versus people without HIV naive, you'll see this shift where you have higher levels of these glycolytic genes and lower levels of these oxidative phosphorylation genes. When you compare naive people with HIV to people without HIV naive, you see the same shift. It's suggesting an induction of glycolysis. But then when you compare the moderate users with HIV to the naive people with HIV, you see it flips. where you have more oxidative phosphorylation, and the same with the daily users. So this is consistent with what I just showed you, where you had more mitochondrial activity in the monocyte-derived macrophages. So next, we did the same sort of thing with inflammatory genes. The inflammation story is a little bit different in people without HIV versus people with HIV, which I think is consistent with the data that we showed before. But the overall idea, I think, is if you compare daily users of people with HIV to the naive people, sorry, without HIV to with naive without HIV, you actually get a reduction in most inflammatory cytokines. But if you compare people with HIV, this is a word salad, to people without HIV naive, you get more of these inflammatory cytokines like IL-6, IL-1 beta, CD68, and less of the anti-inflammatory. But when they use cannabis, this flips, which is, again, consistent with what we and others have shown in previous studies. So as a somewhat of a proxy for these changes in metabolic activity, we looked at GDF15. GDF15 has been getting some media because it is this mitokine and it responds to mitochondrial stress along with FGF21. And we measured it in people, in the brains of people with HIV, and we found that it is elevated in brain tissues of people with HIV with cognitive impairment. And this is in a previous study led by Dr. Bustani in the lab. And so we thought, if this is true, then the people, the serum of these people that we're studying, they may, if they're not using cannabis and they have worse cognitive impairment, they may have higher levels of GDF15, and maybe it'll go down with cannabis use. And that's what we found. You don't see higher levels of GD15 in this is GD15 on the y-axis, and then people without and people with HIV on the x-axis, as we've seen a few times before. You don't see much difference between people without HIV using cannabis. But in people with HIV using cannabis, you see people with HIV not using cannabis, the naive, you see high levels of GDF15, and it is reduced as in the people that are using cannabis daily, suggesting that there may be less mitochondrial stress in these people using cannabis. So I don't have time for this, but I'm going to say it real quick. What we've done most recently is we've taken 30 MDMs RNA from 30 different MDMs, so 6 groups, and we took 5 from each group, and then all the treatments for each MDM, and we did RNA sequencing, and we found some things that I think confirm what people are thinking in HIV. This is just to show you that we have people that can do all the fancy analyses. This is very preliminary as far as we want to do a lot more, but when we looked at things on the right side, you can see these hallmark inflammatory responses, especially involving TNF alpha, are very significantly upregulated in people with HIV. And when I just looked at a few transcripts, we literally got this like 2 weeks ago. But when I looked at just a few transcripts that are important, like TNF alpha, IL-1 beta, hexokinase, which is important for glycolysis, DUSP1 is important. I can't remember why. And then this is PTGS2 is this cyclooxygenase, plays a role in inflammatory responses. My overall point is the blue is people without HIV naive. The red is people with HIV naive, and the green is people with HIV using cannabis. I group moderate and daily. You see this over and over that you get this increase in people with HIV, and cannabis is moderating it. We are also looking at protein in 81 of the samples using this fancy Illumina machine that is supposed to measure 10,000 proteins, and we're waiting on those data. So overall, I think our hypothesis, what we found so far, it's consistent with the hypothesis that cannabis may be anti-inflammatory in some people with HIV, and what we think these data will allow us to do, and there probably are other cells that are just as important, but to identify the people that could be amenable to such therapies, there's a whole bunch of questions, and then know that we have time to do that. And then we have the obvious limitations. I'm sure y'all can think of others. And lastly, again, just have an awesome group in my lab, and then a really great group of people at UCSD and even beyond that we work with. I probably miss people, but thank you all very much.
Thank you, Adam. That was awesome. It's great to see your work and to see how it's kind of evolved over time. I think there are some questions in the chat. I think Jonathan, for anybody who wants to unmute themselves and ask their question. My question was already answered in the chat, so you're good there.
I can look at the chat or I don't know how y'all want to do that.
Jonathan Geiger had a bunch of questions about lysosome function.
Okay. All right. Let me get to, let's see. Oh, yeah. Rachel Schreier, urine tox screens are done at each visit. That's very important. There's so much data and moving parts. We've got
It's just because we work in with substance use as well, and it's very tough, right? Because you're trying to focus on a single substance, and there's all of these other factors, right? So even if they may be toxin negative, they could still, I don't know. Have they been maybe? I don't know if you guys did structural clinical interviews or CDs to kind of have that history or.
Yeah, and they are there, and I'm not the expert on everything it's done, but I do know we're looking at route of administration, which we tried very hard to just limit it to inhalation. And they are asking them a lot of things like how long they've used. And then we are doing urine toxicology. So all that is there. It's not included here. And those data will come together, but hopefully for like really when we cap all this stuff off. So Jonathan, Dr. Geiger, TREM2 mutations affect lysosomal function, and decreases in TREM2 have been associated with decreased cognitive function. Any evidence of disturbed lysosomal function? We are looking at that, and we have started. I mean, the problem is we would love to do it all. The problem is the limited number of cells. Is somebody saying something?
No, that was me. I was just asking.
Okay.
Because again, I think there was some data where you had people with and without HIV that were on, kind of like that 2x2. And do you find it easy or hard to find, uninfected individuals that, it's very hard to find comparison groups. And so how similar are those other groups in terms of all of the other factors? They're probably similar if you're looking within HIV with and without, but you're comparing it to uninfected with and without, and they're likely not to be the same. So I just didn't know because, just the characteristics of those groups.
Yeah, and I can't speak to that too much. I know we try to, like GDS, for example, and I hate to delve into this, but it controls for age, for example. And then we try to control for age. We run tests to make sure that age and things like that aren't affecting the outcomes, or at least aren't related too strongly to them. So yeah, we try to take all that into consideration. And so far, we haven't found anything that is overwhelmingly related to the outcomes to it, and limiting, or they aren't. What am I trying to say? Reduce the impact of the associations that we found. Let's see. Another link to lysosomes is lysosome damage, particularly rupture of induced leakage of cathepsins, acts as a critical damage associate, activates the NLRP3. Okay, that's good. Yeah, we have a lot of things going on. Does anybody want to read what they have? Any questions?
I think Becca had a question. She's up in the corner.
Yeah, fantastic talk. Also a big fan of the inflammasome, and I absolutely love all of that work. And I was wondering specifically when you looked, it was kind of counterintuitive that your IL-18 RNA was higher in the CBD beta treated, but then your protein was lower. And I know you can't look at IL-1 beta protein because you're treating with beta, but have you considered looking at other things that are often secreted, like LDH for pyroptotic death, or caspase-1, or even gasdermin D, as other markers of whether or not those cells are just dying and you're losing your protein signal? Or
Yeah, that is a good point. I mean, we're doing in the cells, we haven't. I mean, I have thought of it. It's just comes down to like having the money to, because it can, by the time you treat with 6 treatments and then you have 120 patients and then you have triplicates of each treatment, like there's only, and then an ELISA costs $500, or you can do mesoscale, which costs 3,000. You know what I mean? So yes, we've thought about it. I would love to send all the samples to one of these like Olink, or like we sent plasma to Illumina because they said they could give us 10, and they gave us a deal. And they could look at 10,000 proteins. So yes, I think we need to look at all that. It just comes down to money always, and then limited. You know, you get your blood. You can only take so much volume of blood. So how much supernatants do you have? But those are good things to think about. Like I didn't think about looking gasdermin D in the media necessarily, but I guess you should because it's secreted.
yeah,
But
and
yeah, there.
it just might shed some light on to whether what's happening, and if they're just being primed to produce 18 but not then secreting the 18, why aren't they secreting the 18? It just was very interesting.
It is, and then there's the idea that transcripts don't always line up with the protein. For some they do, some they don't, and the 18 it really acts weird because you see this stair step change based on cannabis use, and it's different in people with HIV and without HIV. So these are good questions that we're going to have to get a little bit more granular and mechanistic to try to figure them out. But there are this studies. I mean, that's the problem with, not the problem, but it's one of the benefits of human studies. They always open up more questions. And you're like, the further you go, it's the onion analogy, right? The further you go, the more layers there are.
And I think you work in a very similar style to the way Rebecca and I work, right? So you've got Jenny at doing kind of the imaging clinical. You've got, you at the kind of wet lab doing the blood-brain barrier. It sounds like a very similar model, and I think it's a great way to address the questions because you're able to kind of pull it all together. We'll have to bring Jenny and Cecilia. You'll have to come in, like, talk about these other pieces as things evolve. Scott, do you want to? I know we're at the hour. I think Scott was asking about the effects of exercise. Here comes Dr. Latender.
Yeah, hey Adam, great job. Is Jenny here? She texted me that she's on her way to INS in Philadelphia, and so I wanted to make sure, since her name was mentioned, she asked me to let everyone know that she's in a taxi, so and was having difficulty connecting. But my question, Adam, when you were presenting the mitochondrial data, it just made my unusually wired brain think of exercise. And so I was wondering if there were any consistencies between what you were seeing with mitochondrial signal and cannabis with mitochondria and exercise, if you know anything of those data.
Well, we are looking at it in Ron Ellis grant. We just haven't looked at macrophages in that grant. So I'd have to think about how best, what we're seeing. I mean, I think it's consistent. So insofar as like cannabis is inducing this more M2 leaning phenotype metabolically, and I would assume that exercise does the same in people with HIV. What we've seen so far in the study we're doing with Ron Ellis, we're looking at like this GPLD1 with exercise and how it affects the brain. It does suggest that exercise is improving mitochondrial function in neurons and astrocytes, but I can't speak for macrophages. But that is something we want to look at. But yeah, it's a good question. And I will say, maybe, this gets to my point about how, not everybody's the same. And some people, probably everybody could use exercise. Not everybody could use it, but exercise is probably, as long as you don't do too much of it, it's probably good for everybody. Whereas cannabis may not be that way, but you may be able to partition the people that could benefit from one thing or the other. And I shouldn't say even cannabis. Like if we can figure out cannabinoid receptor agonists, very selective ones that limit the downside effects of cannabis and promote the positive effects, that would be great.
It strikes me a lot of people with HIV as they get older, and a lot of people without HIV as they get older have various pain syndromes, and that a little bit of cannabis might have some analgesic effects that would enable them to exercise. So I don't know if any of us has already submitted an application to look at the combined effects of moderate cannabis use and people's ability to exercise when they have mild to moderate pain syndromes, but just might be an interesting thing to do and impactful. So
yeah, that's true. That's interesting.
I'm gonna say thank you, Dr. Fields, because I know we're a little over the hour, and I get to jump on a substance use meeting. So
Thank y'all for inviting me. This was great.
Caroline, do you want to? I think it's Eileen Scully who's speaking next week.
Yeah, next week is Eileen Scully from Johns Hopkins, and her talk is exploring the impact of sex steroid hormones on HIV pathogenesis.
So super excited to have her, Adam. Thank you so much. And Adam and Scott, please tell Jenny. I mean, we're going to be getting you guys for next year, so I'm going to have Caroline not jot that down because I would love to hear at the different levels how it all kind of pieces together. But happy Wednesday, and see y'all next week.
All right. Thanks all.
Thank you all.
you.
Bye.
Bye.